I'm going to mention diffusion first because it's probably the transport process that we're going to see the most examples of in our course, especially in this first half. Now, diffusion is due to the random movement of molecules known as Brownian motion. The identical videos on the left and right side of the slide show a bunch of molecules in solution that are moving by Brownian motion. And you can see that they're bouncing around all over the place. The yellow highlight is tracing the path of just one of those molecules, and you can see that it appears to be moving randomly in all directions, and it's bumping into other molecules along the way. The people who study this stuff sometimes refer to this trajectory as a random walk. I feel like I've been taking a lot of random walks over the past couple of months. But the phrasing is actually really helpful because one of the key things about Brownian motion is that the trajectory of the molecule is non-linear. The molecule doesn't move in a straight line. And so the distance that it travels is not in equal proportion to the time that it spends moving around. In other words, diffusion isn't like running a marathon, where every single step takes you closer to the finish line. In fact, running a marathon using Brownian motion would give off tons of chaotic energy and probably take you your entire lifetime. And it was actually Einstein that figured out that you could describe diffusion using an equation known as the root mean squared equation. And the equation tells us that time and distance have a non-linear relationship because the distance a particle diffuses increases as the square root of time. One of the things that makes the situation inside of our bodies just a little bit more complicated than what you're seeing in the slide animation here is that we're three-dimensional. So when our molecules move randomly, they're moving left and right in the x-plane, they're moving forwards and backwards in the y-plane, and they're also moving up and down in the z-plane. And with each of those dimensions, diffusion gets less and less efficient with respect to time. Now, the point of showing you this isn't to try to scare you with a whole bunch of fancy math. It's really just to emphasize that it's really inefficient to move a long distance using diffusion. The most likely scenario is that you're going to take a large number of steps and you'll just end up pretty close to where you started at. If we think about diffusion in terms of a population of molecules instead of an individual molecule, then we could summarize the whole process by saying that it's passive and based off of random molecular movements. We could say it's very slow over a large distance, but extremely fast over a short distance. If I only need to take a single step in one direction, then even moving randomly, I would get there pretty quickly. Diffusion rate also increases when temperature goes up. So you know from your chemistry lab that when you want to get out of there quicker, you can make your reaction go faster by heating it up. And that's because heat increases molecular movements. Diffusion can and actually does happen everywhere, and it can happen in an open system like a beaker or inside of a cellular compartment. It can also happen across a divider or a partition like the plasma membrane. Larger molecules tend to diffuse more slowly than smaller molecules. And most importantly, to observe a change in a population of diffusing molecules, there has to be a difference in the concentration between two places. So in other words, it requires a concentration gradient. The term concentration gradient is really important, especially for our upcoming unit on membrane potential later in the course. It's not really a difficult concept, but it is something that's really easy to get confused about. So let me say it again. The concentration gradient is the absolute difference in solute concentration between two places. It's actually something that you can calculate. So in the leftmost image on this slide, we start off with 10 red molecules in the left compartment and 0 red molecules in the right compartment. This means that there's a concentration gradient, or difference in concentration, of 10. The next panels show molecules moving by diffusion, until there are 5 molecules on each side. From that point on, the concentration gradient is 0. Now, you'll also hear people, me, using the term flux when describing about the movement of particles. Flux describes the movement of a particle in a single direction. So if you like physics, then you could actually call flux a vector. Because the molecules are moving randomly, there's going to be flux in every possible direction, left, right, up, down, forwards, backwards. So flux is really a term that describes each step in Brownian motion. But thinking on a larger scale, if you added up the flux from every molecule moving at the exact same time, you would be able to see an overall pattern of the movement for the population. And that's known as the net flux. So for example, if you had more molecules fluxing left than right, the net flux would be left. If you had more molecules fluxing right than left, then the net flux would be to the right. And if you had the same number of molecules fluxing left and right, have to be really careful not to get tongue-tied here because these recordings are going to live into oblivion. So in that situation, the net flux would be 0. And that's the situation that exists at equilibrium, like we saw in the rightmost beaker on the previous slide. So if anybody still gives fluxes about flux, then I should probably also mention that we can calculate the rate of diffusive flux for any molecule because it's equal to that molecule's diffusion coefficient times its concentration gradient. Now, diffusion coefficients are always constants and they're going to be unique for every molecule, so we don't really worry too much about them. Instead, the primary determinant for us is almost always going to be concentration gradient. So make sure that you spend some time thinking about and applying that term. Now, things get a little bit more complicated when we have diffusion across a biological membrane, and that's because of those unique properties of the membrane that we were talking about a little bit earlier in the lecture. This diagram shows you all of the factors that can influence the movement of molecules across biological membranes. The pink boxes represent terms related to diffusion, and the blue boxes represent terms related to permeability. So in order for a molecule to move across a membrane passively, it has to first be permeable, and then it has to diffuse. We're going to practice applying these terms, and we'll talk about how each influences diffusion when we're together in class. One of the things that often surprises people about this course, and especially this unit, is how much we talk about diffusion and other molecular processes. But the reason that we do this is really because of structure-function relationships. If we think about structures inside the body that really need to maximize their rate of transport using diffusion, we often see that they've optimized those parameters that I just mentioned. Now, one example that we're looking at here is the lungs. If you take a look at the internal structure of the lungs, like in the left image, you can see that they're highly vascular. They have a lot of blood flow. By the way, this is another image that was taken from that really cool bodies exhibit, where they actually injected latex into the blood vessels to create this image. Now, having all of this vascularity is a way to increase the surface area. The same is true for the small intestine, which you're seeing in the image on the right-hand side. The surface of the tissue isn't smooth. Instead, it's this system of hills and valleys called villi. And that's intended to increase the membrane surface area. If you were to trace the distance across each of the hills and valleys in that picture, you would probably see that it's something like 10 times greater than what you would have if you just measured the width of the image from left to right. So I guess all of this is to say that there is a little bit of method to my madness, and I'm not trying to waste your time. This actually is really, really important stuff.